We're going to be moving into the actual DNA structure and function, as the title of Chapter 14 indicates. This will involve going into greater molecular detail as to how DNA is built, how it is copied, because we talked in general about that with mitosis and meiosis. During the S phase of the cell cycle, one of the things that gets duplicated, the primary thing that gets duplicated, is the DNA. So Chapter 14 will talk about that DNA structure and how it gets copied, setting us up for Chapter 15, where we will discover how the cells go about using that genetic information that is in the DNA to create RNA, and how the cell uses RNA to create proteins. This will be our molecular level of getting from the genotype, which alleles are present, those variations of the gene, and the gene is the DNA sequence, to the phenotype, the actual characteristic that is able to be seen. Chapter 14 strictly deals with the genes. How do we make copies of DNA? A little preview here of things to come. There's Dolly the sheep, the first large mammal to be cloned. That technique we will go into greater detail in a later chapter, but essentially a mammary gland cell, which is a non-sex cell, it's called a somatic cell, was taken from one sheep. The nucleus was removed from that cell. And what was put instead was the nucleus, the nucleus was taken out of that mammary cell, and that nucleus from a mammary cell was placed into an egg cell from a different sheep. And that egg cell had had its nucleus removed. So we have an egg cell with no nucleus receiving the nucleus from a mammary gland cell. The mammary gland cell is diploid. It has pairs of chromosomes. And that having pairs of chromosomes was a signal to that egg cell cytoplasm that the egg had been fertilized. Because when an egg gets fertilized, a haploid egg receives the genetic material from a haploid sperm, and that first cell, the zygote, is the first diploid cell. So an egg cell that received a diploid nucleus was tricked into thinking it had been fertilized, and then that cell, that egg cell that believed it was fertilized, began to undergo mitosis, and that was implanted into another sheep's uterus and grew into the lamb that became Dolly. Frederick Meichner is listed here. Again, you don't have to memorize the names, but he is the one who discovered nucleic acids. They are called that because we knew about the nucleus of cells. If you take cheek cells and stain them, there's a dark blue dot in the middle. And it was called the nucleus, which was Greek for meaning kernel or center. So these nucleic acids were acidic molecules that were extracted from the nucleus. At that point, even though we had molecules that had an acidic pH, these nucleic acids, it was still not known what their role was in the cell. So there are additional experiments done to determine what the role of that molecule was, and there was also interest in determining what is the genetic material. We could follow genetics based on Mendel's and experiments from others, but it still was not known what the genetic material was. Dr. Griffith, these poor little mice, were subjected to experimentation with an organism called Streptococcus pneumoniae. As its name indicates, it can cause a fatal pneumonia. There were two variants. The S strain is S for smooth because when these bacteria are grown on an agar surface, they're very shiny. They have a smooth surface. There's another variant of this exact same organism, Streptococcus pneumoniae, but when it is grown on agar, it has a very rough appearance. It's matte, it's dull. And when those rough bacteria are injected into an animal, they do not cause disease. Well, this poor little mouse received a heat-killed variant of the smooth strain and survived. So there was something about, you would think, dead bacteria, well, they can't cause any problem. However, if you have the heat-killed S strain, those bacteria are dead. If you inject them into an animal, nothing happens. But they also provided live, and that should be virulent, ENT, non-virulent, meaning unable to cause disease. So the rough bacteria were alive. They mixed them with dead, smooth bacteria and let them set for a while, and then injected that mixture, poor little dead mouse. So Griffith's conclusion was that there was some chemical, some aspect of those smooth bacteria that had to be chemical because the bacteria were not alive. Some chemical inside those smooth bacteria transformed the rough bacteria into smooth bacteria that killed the mouse. This is a little more detail on that experiment. So if we have the control where we have the rough bacteria, we inject them, have a healthy mouse. The heat-killed smooth bacteria alone, just the heat-killed smooth bacteria, inject them, well, dead bacteria don't cause disease, healthy mouse. But if we had live rough bacteria plus dead smooth bacteria, and these are representing the rough ones, these are representing the smooth ones, but they're dead, inject them, oh dear, poor dead mousey. And then they looked at this poor little dead mouse and extracted blood and grew that on agar, and it was a smooth bacteria. So if you have the rough bacteria and the dead smooth, you will get smooth bacteria that are still capable of killing the poor mice. And we know now that the smoothness, that shininess, is because of an extracellular capsule, a slimy polysaccharide layer. And there's something about that slimy layer that increases the bacteria's ability to cause disease. The bacteria that do not have that slimy layer don't cause disease. And that slimy capsule layer most likely hides these bacteria from the immune system so that they can get into those lungs and secrete their chemicals that cause illness. And the rough bacteria do not have that, and they get recognized by the immune system. The follow-up to that was, okay, some chemical in the smooth bacteria transforms the rough. What chemical is it? Well, we have a test tube where we have heat to kill the smooth strain, and then the capsule components are removed from the solution. And the little green dots represent proteins, blue represents DNA, red represents RNA. So if the DNA and the RNA and the proteins are present, we add the R cells and they get transformed into smooth cells. So that says, well, it's the protein, the DNA, or the RNA. We don't know which one. We can chemically and physically remove the protein so that RNA and DNA are present. If both nucleic acids are present and the protein is absent, we can add the rough cells and they get transformed. So it's either the RNA or the DNA. So if we take this one and we digest it with nucleases that specifically destroy RNA, so this tube has protein and DNA. We add the rough cells and they are transformed. But if in this test tube we use a separate enzyme that instead destroys the DNA, but the proteins and RNA are still present, add the rough cells and there is no transformation. So that was the confirmation that transformation requires DNA, not just any nucleic acid, specifically the DNA. An additional experiment showing that DNA is that genetic material was done with a virus called a bacteriophage. Bacteriophages are viruses that infect bacteria. So yes, even bacteria can catch a virus. This particular virus has this black line representing its protein coat called a capsid, and it has nucleic acid, and it only has DNA. Viruses, unlike cells, have either DNA or RNA. They never have both. This virus, this bacteriophage they used, has only DNA. They did an experiment where they added a radioactive element. And in one experiment, only the phosphorus was labeled with radioactivity because DNA and RNA have phosphorus and proteins do not. This entire pathogen, this virus, has only protein and DNA. There's no carbohydrates, there's no RNA, there's nothing else but protein and DNA. So if the phosphorus is labeled with, if the phosphorus is radioactive, got it, radioactive phosphorus, as the viruses duplicate inside the cell, the DNA will become radioactive. In a separate test tube, they labeled the sulfur with radioactive sulfur, 35S. And that means only that protein coat will get labeled because nucleic acid does not contain sulfur. If the virus replicates, only the outer protein coat will be radioactive. They mixed in one test tube the radioactive phosphorus viruses with bacteria to allow infection to happen, mixed them all up, and then centrifuged them, and that will separate out the phage from the infected cells. The virus particles are very tiny. They will float. The infected bacteria are heavy. They will sink. And what they found is that the bacteria had radioactivity and the fluid portion did not have any radioactivity. So the nucleic acid exited the virus, entered the bacteria, and the protein shell is left over, but the protein shell did not have any radioactivity. Only the nucleic acid did, and so the nucleic acid entered into the bacteria. In the protein experiment, the same thing. They allow the viruses to infect the bacteria as they normally do, mix it up, and then spin it so that the empty protein heads leave and the infected bacteria sink. And in this case, the bacteria had no radioactivity, but the fluid containing the protein shells did. So that was another piece of evidence that the genetic material was the DNA that entered the bacteria and then directed those bacteria to create more viruses. Get me out of the way. A reminder of our nucleotide structure we talked about way back in our macromolecules chapter, that a nucleotide is made up of a sugar. If it is RNA, it will have hydroxyl functional groups of both the second and third carbons. If it's DNA, which is deoxyribose, it will have a OH at the 2' region, but it will have an H at the 3'. So if we have just the sugar and the base, that is a nucleoside. With one phosphate, it's a nucleotide monophosphate. With 2, it's a nucleotide diphosphate, and with 3, it's a nucleotide triphosphate. The base refers to one of these molecules that's added to give it its identity. That is the R group. Just like with amino acids, we have the R group that provides the true identity of each amino acid. The base, which has a carbon and nitrogen ring, is what gives the identity to the nucleotide. We have two groups, the pyrimidines, which each have a singular ring, and the purines, which each have a double ring. DNA has cytosine, thymine, adenine, and guanine, and RNA has uracil, cytosine, adenine, and guanine. The C, A, and G is exactly the same in both nucleic acids, but only DNA has thymine and only RNA has uracil. DNA and RNA are extremely similar chemically. The sugar has just one oxygen difference at the third carbon position. That's it. And between a thymine and a uracil, the difference is this methyl group. Thymine has a methyl group on the second carbon, and uracil just has a hydrogen. DNA is also double-stranded in cells, and RNA is single-stranded in cells. A little bit of history. This is an image on the left of Watson and Crick with McCarty, who won the Nobel, Watson and Crick won the Nobel Prize for discovering the structure of DNA. They started out building molecular models. If you did that in your general chemistry class, you know that it's pretty complex to get each atom with the right number of bonds in the right order to build a molecule. So you can imagine just building one nucleotide is extremely complicated, and then trying to figure out how one nucleotide joins to the next, additionally complicated. And add to that the fact that when they started building these models, we knew the structure of a nucleotide. That had been discovered, but we did not know the overall structure of DNA. Is it double-stranded or triple-stranded? How are the strands held together? That was not known, and that is what Watson and Crick eventually figured out. The picture on the right is an X-ray crystallograph that was done by Dr. Rosalind Franklin. That helped them to figure it out. Dr. Franklin did not get the Nobel Prize because she died before the prize was awarded, and Nobel Prizes are not awarded posthumously. There is controversy regarding how did Watson and Crick obtain that X-ray crystallography that, since they knew how to read them, told them it was a double helix. The articles I have found that the data wasn't particularly stolen, but they weren't exactly honest with how they obtained it. Should have a link to an article. I'll get it in here. From The Guardian, they indicated that there wasn't driven by sexist disdain because Watson and Crick would have probably done the same way to get the data if a male scientist had produced it. But later in that same article says, had Watson bothered to take notes during her talk, because Dr. Franklin had given a talk about this image and what it meant for the structure of DNA, if he had taken notes instead of musing about her dress and her looks, maybe he would have gotten that numerical data that was critical that they ended up finding by obtaining her picture 15 months earlier. DNA is often shown as this little spiral staircase, and it is essentially that. This is looking at if you built your ball and stick models to get the double helix made. And this is what is going on when we look at all of the atoms that are actually involved. So here is a sugar. There is the phosphate. So we call it the backbone. It is the phosphate sugar backbone because we go phosphate to sugar to phosphate to sugar. That is where the covalent bonds between individual nucleotides are made. So all in yellow is a thymine nucleotide. It is covalently bound at this position to a guanine nucleotide. We have these numbers, 5 with a hash and 3 with a hash. Those are pronounced 5' and 3'. They refer to the number of the carbon in the sugar ring. So this is carbon 1, 2, 3, 4. This is carbon 5. The 5' end, where we have no more additional nucleotides, always has a phosphate functional group. And the 3' end always has a hydroxyl functional group. The structure of DNA is not just a double helix, it is also called anti-parallel, because we see on this strand the 5' phosphate is facing up, but on this strand the 5' phosphate is facing downward. We know that's the 5' end because there's a phosphate on it, and we go phosphate, sugar, phosphate, sugar. There's the 3' end, it has an OH. This has to be the orientation because it's the only way that we get this even, same width all the way along the molecule, double helix. That's where that X-ray crystallograph came in handy, because it wasn't wide and skinny, because if we had two double-ringed structures, it would get really wide at that portion. And if we had two single ring structures across from each other, it would narrow at that structure. And that's not what happens. It is completely uniform all the way along the length of the DNA. And that is where a feature called base pairing comes into play. Every time on one strand there is a nucleotide containing a thymine base, it will be paired directly across with a nucleotide containing an adenine base. And there are two places between a thymine and an adenine where we can do hydrogen bonds. Likewise, wherever there is a guanine nucleotide on one strand, its opposing nucleotide will be a cytosine. And because of their structures, there are 3 places where we can hydrogen bond. Covalent bonds hold a single strand together, the bonds between individual nucleotides. But between this strand and its partner strand, it's only hydrogen bonds. Hydrogen bonds are about the weakest forces, but we have billions of nucleotides, and we have a minimum of 2 hydrogen bonds between each nucleotide pair. That means we have multiple billions of hydrogen bonds holding our strands together, and we'll see later that the DNA also compacts down. This is an image showing what a sequencing reaction looks like. DNA sequencing. We do have techniques where we can figure out the actual order of the nucleotides in a DNA sample. I'm going to have a video in the next slide that will talk about having to make copies of that DNA so that we have enough DNA to be able to get the sequence. And that is definitely true. The video talks about chopping up the DNA that you're interested in and using molecular techniques, sticking it into a circular piece of DNA called a plasmid. We do that because we have previously determined the sequence of the plasmid, and we can do the sequencing. There are other ways to accomplish making copies of DNA, namely through PCR, the polymerase chain reaction. We will talk about those cloning techniques and PCR techniques later, but know for now that there are ways of getting multiple copies of the DNA to be able to do sequencing. What this is showing is dye-labeled di-deoxynucleotides. And I did misspeak the, here we go, now get back to it, I'm getting dizzy. It is the 2' and the carbon number 2 that has only a hydrogen in DNA. We have to have this OH because that is the position where we do our dehydration synthesis. So the 2' carbon has an OH in ribose, so in RNA, and the 2' carbon has only a hydrogen in deoxyribose, or DNA. For our sequencing, we not only have the deoxynucleotides, which is what we need to build a copy of DNA, we also have di-deoxy, meaning there's a hydrogen at that 2' carbon, which is what should be on a DNA nucleotide, but there's also only a hydrogen at the 3' end. If there's only a hydrogen at that 3' carbon, then we cannot build any more nucleotides on that. It will terminate the replication. It will terminate the chain. So Sanger sequencing is also called chain termination. We essentially do DNA replication in a test tube with all of the regular nucleotides, the deoxynucleotides, but we also, in each tube, have one di-deoxynucleotide that has a fluorescent label on it. And when we have a di-deoxynucleotide, the DNA replication will end, and wherever it ends will be that di-deoxynucleotide that is a particular fluorescent color, and we run it through a gel with a laser that will show us the sequencing. This is our normal deoxynucleotide. We have a hydroxyl group at the 3' carbon and an H only at the 2'. But in a di-deoxynucleotide, they both have hydrogens. It does work. The DNA polymerase is the enzyme, ACE enzyme, that builds a polymer, and the polymer it's building is DNA. It will need the DNA we want to know the sequence of to be able to have something to read, to make a copy of it. It needs the nucleotides. Those are the individual monomers to build our polymer. It has the modified ones that will terminate the length. And we also need this thing called a DNA primer to prime, to get the reaction going. So it is a short sequence of known DNA that will match into that plasmid in this case, so that the DNA polymerase has a place to bind and then extend into the unknown sequence. And I'll let you continue to watch that, and you can watch it as often as you need to go over that sequencing process. But it does go through the whole process of the reaction. It goes through how the reaction goes through a gel and how the laser reads that to interpret it to the sequence of the DNA that we want to know the order of the nucleotides. We can also separate DNA on the basis of size using gel electrophoresis. It's going to be our lab this week. We're going to do a DNA extraction. We are not going to do the electrophoresis. It requires more equipment than we've got at home. But DNA, because of its phosphate groups, is negatively charged. And if we put it into a gel matrix and put that in buffer and provide an electrical charge, if we put the positive electrode at the end of the gel, all of the DNA pieces will be attracted to that positive end because all of the pieces have a negative charge because of their phosphates, and then they separate based on size. Gel meshy network, little pieces migrate really quickly, big pieces get caught up in the gel. So these two lanes contain fragments of known size. The large ones are so large they kind of smear, but then they start to separate out, and the smallest ones are toward the bottom. And then these would be the experimental samples, where we have in each sample, it looks like one smaller and one larger fragment. We can do a graph of the known size of these bands versus how far did they migrate. And then based on how far each of these bands migrated, we can determine their size. We know from our cell structure lab and lectures that eukaryotes have a true nucleus, and that is where their DNA and RNA are located. Whereas prokaryotes do not have an internal compartment. The nucleoid is just wherever their DNA is sitting. Eukaryotic DNA, even more so than prokaryotic DNA, has to compact down or it won't fit in our cells. So our double helix, shown relaxed, it is actually representing this little tiny piece of a chromosome where DNA is wrapped around histone. Histones are proteins that our DNA wraps around. This is representing just this tiny little piece where these yellow discs are the histone proteins, and the blue is our DNA that wraps around the histones. It continues to condense into chromatin after replication, so that when we look at our cells in prophase of either mitosis or meiosis, we see those condensed, duplicated chromosomes. Prokaryotic chromosomes also condense, but they do not have histones. They just coil in on themselves to compact. DNA replication happens by a mechanism called semi-conservative. The original concept was that there are 3 hypotheses. It could be conservative. We know DNA is double-stranded, and that somehow it is copied to make 2 new strands that hang out together, and the 2 original strands hang out together. It could have been dispersive, where during the copying, each molecule is a mixture of original and new DNA. We now know that the actual way that DNA is replicated is by a means called semi-conservative. The DNA strands are held together by hydrogen bonds. Enzymes break those hydrogen bonds so that each strand is separated. It doesn't completely come apart and fly around, but it is separated, so we have single-stranded DNA. And DNA polymerase will read each of those strands so that the result is one old and one new strand every time DNA replicates. One way that we had evidence for this was, once again, using radioactivity. DNA contains nitrogens in our nitrogenous bases, and this time we used 2 different forms of nitrogen. The standard nitrogen with an atomic weight of 14, and an isotope of nitrogen, nitrogen 15, that has an additional neutron. Right? We increase the molecular or the atomic weight, we are increasing the number of neutrons. What they showed was if you grow the E. coli in heavy nitrogen, and then into the light nitrogen. Of course, the heavy nitrogen DNA is going to be heavier. You start out with 100%. They are only provided with nitrogen that was the heavy isotope. That is where it settles out when you centrifuge it. If you undergo one round of replication and then isolate the DNA, it is all at an intermediate level. But if you let this undergo another round of replication in the presence of the 14 nitrogen, half of those resulting strands are at that intermediate weight and half are lighter. And as you keep going, the proportion that are the heavy nitrogen are fewer and fewer because we only had the 15 nitrogen at the original duplication. Sorry if that was an abrupt transition, but my Instacart grocery delivery arrived. So we end up with less and less, a lower and lower percentage of the heavy nitrogen DNA because we only did one replication in the presence of that heavy nitrogen. Every subsequent replication was only in the presence of the 14 nitrogen. And that was what provided the evidence that our DNA replication is semi-conservative. We conserve one original strand but get one new strand every single time that DNA replicates. This image does contain all of the components that happen in DNA replication, and it's minimally useful because it is static and DNA is a dynamic process. But I will go through what each of the enzymes are doing, and then once again have a video that takes you through dynamically what's happening at each stage. Our double-stranded DNA, and if we follow each strand, this one is 3', and when we get to the opposite end, well, it should be 5'. I'm not following it. Yep, that one's 5' and we end up at 3'. Good. We have to open up the DNA in order to make a copy of it. The enzyme that opens the DNA is helicase. It unzips the helix. When we do that, if we just pulled open a twisted strand, it would get completely crumpled up behind that enzyme. We have this green ring representing topoisomerase. So an enzyme interacting with an isomer, and topo is like topography. Have you ever heard of a topo map? It's a map that shows you the elevations. Well, the topoisomer is relaxed DNA versus crumpled up. So the topoisomerase prevents the DNA from getting twisted up as the helicase breaks the hydrogen bonds. These purple dots represent single-stranded binding proteins because they are proteins that bind to the single strand. The reason for that is if we didn't do something to kind of keep the single strand separate, they would hydrogen bond right back up. Those single-stranded binding proteins prevent DNA from immediately zipping back up after helicase breaks hydrogen bonds, and it also keeps the DNA stable and protects it from being damaged by other enzymes. Now, in order to make a copy, we do need DNA polymerase to build a polymer of DNA. We actually have two different ones, and we'll get to why in a moment. We have a leading strand and a lagging strand. They are called that because the leading strand, as it appears here, is synthesized continuously. Once the DNA polymerase recognizes the template strand, It provides the bases that complement or base pair with the template and build the new strand. DNA polymerase 3 is the enzyme that works on the leading strand. And the DNA polymerase 3 requires what is called a free 3' OH, that 3' end of the molecule. This is our parent strand, and when we look down here, yes, we had a 3' end that was available for DNA polymerase to bind and then synthesize the new strand. On this one, and it is labeled wrong, that should be, let's put that in black. This is the 5' end, and this is the 3' end, right? Because of the, yes. Get me back to the arrow. There. The original strand had a free 5' end. DNA polymerase cannot recognize that phosphate. It's a very large functional group. It should recognize a hydroxyl, a very tiny functional group. So in order to get a 3' hydroxyl, we have another enzyme called primase. It's an enzyme that's going to create a primer. And it's going to be RNA. Why? We don't know. It just is. It's an RNA primer. A short stretch of RNA that can base pair with the parent original DNA strand, and it is synthesized in a 5' to 3' direction. That's what provides us a 3' OH. And then DNA polymerase 3 will bind to the end of that primer and make DNA that is complementary to the apparent strand. And then we have to wait for the helicase to open up the DNA a little further, and then RNA primase will back up, make a primer, DNA polymerase 3 will bind, and then create more DNA. That is why it's called the lagging strand. It lags behind because we have to open up, make a primer, synthesize DNA. Open up further back, make a primer, synthesize DNA, and keep doing that in a stepwise fashion. Each of those pieces of DNA is called an Okazaki fragment because Dr. Okazaki figured this out. When we finish, then we're going to have pieces of RNA and DNA and RNA and DNA. And we don't want that. That is the role of DNA polymerase 1. It's still an enzyme that will build a polymer of DNA. Its job is to remove all of the RNA primers one at a time. It cuts them out, and DNA polymerase 1 replaces the RNA with DNA. The DNA ligase is the enzyme that makes sure we have all perfect sugar phosphate backbone sealed covalent bonds in the new strand, because to ligate means to seal, to bring together. So ligase seals any gaps between where the DNA polymerase 1 replaced the primer and the Okazaki fragment. Again, I know, static picture, kind of terrible, but at least now you know what the enzymes are doing, so that when you go through this video, it shows the actual process of what's happening on each strand. And so I have this embedded in this PowerPoint, but I also have it separately in this week's module, so you don't have to dig through the PowerPoint presentation to find this. But I wanted to put it in context. This is showing a problem that eukaryotic chromosomes have. our chromosomes are linear. And without having an extension, there is a possibility that the DNA polymerase won't get those last few nucleotides. So the ends of eukaryotic chromosomes have a repeated sequence called a telomere. It's this TTGG, over and over again. It does not encode any product. It is just a repeated sequence. In order to finish in making sure we extend through the end of the chromosome, we have an enzyme called telomerase or telomerase, depending on who you ask. Your question might be, well, why do we care if we copy this junk telomere that doesn't encode anything? Well, it's like a little safety net to make sure that if we do get shorter, if the DNA polymerase forgets or can't get to the end of the chromosome, we have a pretty long chunk of DNA that if we lose it, it makes no difference. So our telomerase has an RNA sequence in it. And we know it's RNA because uracil is in there. It is complementary to this repeat sequence in the telomere. And that is used to synthesize a complementary strand. The telomerase shifts and it's repeated so that we get DNA going back in this direction. So it's a way of creating a primer with a 3' end so that DNA polymerase can make sure it gets the ends of our chromosomes. This is Elizabeth Blackburn, she's a California researcher. She received the Nobel Prize in 2009 for discovering how telomerase works. And it's important because it does show about replicating in eukaryotic cells. There's also a hypothesis that we can only age so much because of our telomeres. If our telomerase gets old or damaged and doesn't function correctly, then our chromosomes do get shorter and shorter, and eventually we're going to lose functional pieces of DNA. And that is called the telomere clock. We went through our DNA replication with the assumption that everything goes right, just like we did with mitosis and meiosis. First we learn it, how is it supposed to go, and then we figure out, what's the effect of having mistakes. DNA polymerase doesn't just go through once, read all 3.5 billion nucleotides in our skin cells and go, yeah, that's good enough. It has a proofreading function, and we should all practice proofreading a little more. Had I done so, I would have seen that the 5' and 3' ends were labeled incorrectly on a provided slide. DNA polymerase proofreads by doing its replication and then backing up. So here we can see we do have good CG, there's a TA, ooh, we're getting some incorrect base pairings. And so if there is an error in the base pairing, the polymerase actually has an activity to cut out an incorrect base pair and replace it with the one that should be present. That doesn't mean it catches every single mistake, but at least does have the ability to do proofreading. We can have mismatched repair. We're going along, and here this strand seems to have been duplicated correctly. We have C's and G's and A's and T's. This one, oh dear, we have a T across from a G. How does the cell know? Is the G the incorrect nucleotide, or is the T the incorrect nucleotide? G and T are not a good match. It should be a G and a C, but the cell has to have some way of knowing which one is the new one that's the wrong one. And that's where these little highlighted adenine nucleotides come into play. We have an adenine with an M on it. That M stands for methyl. A methyl group is a carbon with 3 hydrogens, so a CH3. The process of adding a methyl group is called methylation. During DNA replication, after replication is complete, cells have an enzyme that add a methyl group to the adenines. So that is how the cell can recognize, if it reads the region around this mismatch, the strand that has methylated adenines, that's the template strand. That's the correct nucleotide order. And that would tell it the T is the new one. That's the wrong nucleotide. It needs to be eliminated and repaired. And then that way we end up with the correct process. When we finish replicating, then the A's in the new strand would get a methyl group. We can have damage to our DNA that happens to UV exposure. Ultraviolet light causes an effect called thymine dimers. We should have covalent bonds between the sugars and phosphates in the backbone of each strand. Each base should be covalently bound to that sugar phosphate backbone, but we should have no interaction between bases on the same strand. A thymine dimer happens when we get a covalent bond between adjacent thymines on the same strand. That does cause the DNA to bend. During replication, polymerase wouldn't know quite what to do with that. It doesn't recognize it as 2 separate bases. It'll make its best guess as to what should go in there. The way to repair that is called excision repair. We have enzymes that can recognize that this is not correct and will cut the DNA on either side of that, and a DNA polymerase will go in and repair that, replace it with correctly attached thymines. This is a person suffering from a genetic illness called xeroderma pigmentosa because they lack the enzymes to correct thymine dimers, and that causes lesions and can cause cancer. So these persons would have to stay completely out of sunlight. We will talk more about mutations in chapter 15 when we start talking about how does the cell get from the information of DNA through RNA to proteins. But a mutation is any change in the nucleotide sequence. And we are all almost certainly mutants. The odds that every single time mitosis happened in our cells, nothing went wrong and our polymerase was 100% accurate in correcting any mistakes, we're probably all mutants. We are lucky because sometimes mutations don't have a terrible effect. We could have silent mutations. We should have a GTA here. Instead, we have a substitution. This A gets changed to a T, and we'll talk about translation next time, but when the cell makes the RNA and the RNA gets read, it should have been a valine amino acid, and even though there's a mutation, we still got a valine. Silent. The resulting product of that DNA, the phenotype, unchanged. If instead, in this chunk of letters, we should have had a CCC to give us a proline amino acid, instead we have an adenine cytosine, we get a different amino acid, threonine. That is called a missense. We got an amino acid, it's just not the one we should have had. Depending on where that is in the protein, that may or may not affect its function, but it still doesn't make sense. It's not the right amino acid. Lastly, we could have a substitution of a single base that instead of getting an amino acid, we get something called a stop, a signal to the cell that we are not going to make any more connections of amino acids and the protein ends. That is always going to be harmful. The protein is too short. It will not fold properly. It will not function. So each of those, silent, missense, and nonsense, are a substitution. We have the same number of nucleotides, but one nucleotide gets changed. And if it doesn't affect the amino acid, it is silent. If you get an amino acid, but it's the wrong one, that is missense. And if you don't get an amino acid at all, that is nonsense. It just doesn't make sense to the cell. If we add one or more nucleotides or we delete one or more nucleotides, we get what's called a frame shift. If you look above, we read the DNA in clusters of 3 letters at a time. An A, a G, and a C gives us a serine. A C, a T, and an A gives us a valine. We have to have 3 in a row, and it has to be those 3 in a row to read the frame. If we insert, we put in an extra, let's see, we have an A, C, T, G, T, C. If we insert a nucleotide, we will throw off where that insertion happens. Over here, I guess we have a deletion. They deleted a T and an A. Should have been GTA and then CCC. Instead, we get G, CCC, TAC. Serine is okay. We have the same AGC that we started with, but the valine, we're missing 2 letters, and so the frame we need 3 letters. So it goes 1, 2, 3, 1, 2, 3. And that means we're going to change everything past where that mistake happened. We shift how those triplets of letters are read. Next time we will talk about transcription, how we read the DNA to get to RNA. And then after that, we will talk about translation, how the cell interprets that RNA to build the proteins.